Hello, friends. Today I'll be talking about writing science. And this is important because when you are in science, you have to write, manuscript, you have to give presentation. That's why writing, science and communicating science is very important. So basically, people who have recently joined grad school or PhD or bachelor degree students or master degree students, at some point of their career, they have to communicate science, either giving presentation, talk, or writing manuscripts. So, with all my experience from last 10 years, I have highlighted some of the important points that can be followed while writing a scientific manuscripts. So, while writing a scientific manuscripts, I have highlighted a few of these important points that need to be followed. First, you have to give a title of your manuscript. Then you have to come to the abstract. You have to come material and methods, results, then discussions, then figure and figure legends, main figure legends. Then you have supplemental figures and supplemental figure legends and supplemental tables. Okay, so I will go one by one in each of these sessions. So in today's session, I'll be talking about structuring the abstract. Structuring the abstract is very important for your manuscript because when you submit the manuscript, first the editor will read your abstract. If he feels that this is a very exciting story, then this will go for the review. So that's why it is very important, very crucial to structure your abstract. So there are three kind of abstract people used to do. One is they're used to write, that is called as abstract text. Then you have the graphical abstract. Mostly the self-paced paper, they follow this graphical abstract or video abstract. So I'll go one by one. So let us talk about abstract, how to write the abstract and what is the key points while writing abstract. So when you write an abstract, you should summarize the overall story of your manuscript in a very, very, 250 to 300 words in a very short, and that should convey the important message. That should convey the important message. This is really important. You have to briefly introduce, you briefly write what is your finding, what is the novelty, and how it can change, the science. That you have to highlight in your abstract. Then the editor will find it good, then he will send it for review. So basically, while writing abstract, I used to follow, according to me, I don't want to debate here, but according to me, I follow four different steps. So first step, what I do is, I write, basically, I give a small introduction, and I used to expand the title of your manuscript and orient the reader briefly. So I will just explain it clearly. So here, this is the title of the manuscript. The title is like I have written RNAi mediated reverse genetic screen identified Drosophila sapiens regulating eye and neuromuscular junction morphology. So here the title suggests the identification of Drosophila sapiens and its role in eye and neurons. So basically, when you write an abstract, you have to start with neurons, basic introduction about neurons, then Drosophila sapiens, how Drosophila sapiens are important in neurodegenerative diseases, and what happens when there is dysfunction of sapiens. So that is first few lines or first few sentences you have to write introduction. Then next step is what is not known, and you have to highlight in the text. So here I have written, in the context of sapiens, I have written, while most of the sapiens are expressed in neurons, their functional relevance remains largely unknown. So you have to highlight what is not known. That should come in the second step. And the third step should, how you did and why you did. How you did. So here I have written, like, I have used bioinformatic analysis, then I have identified this many sapiens, I have knocked down these sapiens, I found this phenotype in eyes and neurons, all these things, then a behavioral defect. So you have to find you have to write here what you found while doing this experiment. You have to briefly write it here in the third step. And in the fourth step, you have to discuss the novelty of your research and how it might contribute for further studies. So here you can see that our screen identified a new set of sapiens that regulate I and NMJ morphogenesis. Okay, then what I mentioned here is that the outcome of the screen reported here provides a useful resource for further elucidating the role of individual chaperones in Drosophila eye morphogenesis and synaptic development. So here I have written, novelty, why this is important, and what can be done in future studies. So this is the 4-step. First step, you have to briefly introduce. Second step, you have to highlight what is not known. Third step, how you did and why you did. And the fourth step will be the novelty of your research and how it contribute for further studies or future studies. Okay, so this is, people basically, when they write an abstract, they used to follow these 4 steps. I used to follow these 4 steps. Then comes to the graphical abstract. So here, this is a different example. I have cited a different example. Here, whatever you're finding in your research, you have to graphically mention it. So here, if you look at this image or this research talked about Ross and aging. So here, there are 2 cells. One cell is neuron and another cell is glia. Okay. So here, the glia produce lactate, and lactate transported to the neuron, and because of the excess lactate, it produces more ROS, and ROS induce lipid production, and lipid transported to the glia through this through this receptor, and this is how briefly you have to you're finding. I think in this paper they talked about transport of lactate and production of ROS and lipid, and production of lipid in the neurons, and how glia basically contribute lactate to the neurons, and neuron produce lipid because of excess accumulation of ROS. So that is what the finding in the paper. So this is called graphical abstract. Now another thing is the highlights. So here in cell press paper, they used to make a graphical abstract. Then after that, they highlight the important points. So here in that paper that I shown you before, the graphical abstract, these are the highlights this abstract. Like here written, glia derived lactate fails, neuron metabolism, neuronal lipid production. These are all highlights, important points or main points in the paper. Okay, and finally video abstract. So video abstract is something like people used to mainly convey message through video. So you will come in the video, then you will briefly introduce about your topic, then in brief introduction, then after that you have to rational of your work, then what you find and what is the future direction. You will say in a video the same thing that is written in the main text. You have to make it very short and convey the message in a video, like 1 or 2 minute video. That is called as video abstract. So thank you guys for listening me. So next session I'll be talking about introduction, how to write introduction. Then we'll go over like material, method, result, discussion, all these things. So for today, I'm done. So we'll meet next week. Thank you very much for seeing my video. If you like my video, please like, share, and subscribe.